Hi, year 11s and 12s. This is the very last video for the DNA and proteins topic. I know I said that in the last video, but this is an additional video that's been added on just to cover some content that wasn't covered in those original two videos. In this video, we're going to be looking at protein design, so how humans physically create and manufacture proteins outside of the ones that are naturally occurring in our body. So let's begin. First off, we kind of need to discuss what is protein design. Protein design is when a new protein molecule is created for humans, created by humans, so that we can use it in a biomedical research or medicine or use it for technology. It's basically just a protein created by humans for a specific need. Okay. Now, when we're doing this, we can design these proteins from nothing, so design them from scratch. Or we can take a pre-existing protein and make modifications to it to make it more desirable for what we actually want the protein to do. How this is done is generally scientists will start with what they want the protein to work on. So, for example, if they want it to work on a specific substrate, they then know what shape the protein has to have. So using computer software, they'll determine one, the shape the protein is to take, which will determine the folding the protein needs to have, which will finally determine the amino acid sequence that protein needs to be made up of in order to fulfill this specific need. They'll then, using biotechnology tools, go along and create that protein so they can use it for this specific target molecule that they've designed the protein for. Okay. Once all that's done, the new protein is ready for use. That's fantastic. We've created a brand new protein, but what exactly can we use that protein for? So there's a number of different ways or reasons that we'd create brand new proteins or modify existing proteins for our needs. Okay. First off is enzyme design. So we can create specific enzymes with a very specific active site to a substance we want to break down. Okay. So we use this type of protein design in the food and biomedical industries. We can also use protein design to help study protein and protein interactions. So we can create proteins that are going to interact with other proteins that we know contribute to the development and progression of certain diseases. So we can develop proteins to see how they will work in preventing disease progression. We can develop proteins that will help us make new materials because we can create these proteins to have specific physical properties. So we could increase their strength, or we can increase their toughness, or how ductile they are. We can specifically design proteins for our needs. And we can also use these designed proteins in targeted chemotherapy. So we can design proteins to help prevent tumor growth. And we do this because they interact with specific molecules that are used in the replication of cancer cells. So that's just 4 ways. There's much more than this, but these are 4 ways that we physically use the proteins that scientists are genetically creating. Another example is biosensors. So biosensors are a type of designed protein that can act as a receptor, an antibody, or an enzyme. Okay. What they do is they detect chemical substances in the environment because they bind to those molecules that they're trying to detect, because they are very specific to that molecule. We use them in the medical industry to help monitor blood glucose levels in diabetics. We use them in environmental industries to detect pesticides in rivers, but we can also use them in counter-bioterrorism, disease prevention, food quality, and food safety. If you want to know a little bit more how they can be used in counter-bioterrorism, right up to including food safety, have a look in your textbook. There's a lot more details on those, but you only need a few examples, which is the medical and environmental in this aspect. That's it. DNA and proteins is now finished.